Hi, I'm Carl, and in this video we're going to have a look at questions 106 to 108 of section 3 of the pink booklet. So this is a question about optogenetics, and we're told that this is the interaction between light and genes. Have a read over the paragraph that's given here, but I'm going to highlight two different examples that they've given. So they've talked about two different channels here. So we've got CHR1 responds to yellow and green light, allows sodium ions to pass through, and NPHR responds to yellow light by allowing chloride ions to pass through. Now, then we're told that you can specifically target the location for these genes so that they're expressed in particular cells using something called a promoter. So you can put this into a vector such as a virus, and then it will take the genetic material to the correct type of cell based on what the promoter does. So we'll start with question 106 then. It says, how does the presence of a light sensitive channel protein in the channel membrane activate a neuron? So of course we've got this example here, which is allowing sodium to enter a cell, and when sodium enters a neuron it allows for depolarization, so that will activate the neuron. So question 106, the answer would be A. But I'll go through the other answers. So B says it allows the vector to enter the cell. So the presence of a light sensitive channel doesn't allow the vector to enter the cell. The vector by design should be able to enter the cell on its own, just like a virus. C says it increases the production of synapses. So synapses obviously are going to be these connections between neurons, but the presence of these light sensitive channels will activate the neuron by increasing the production of synapses. And then D says it increases the storage of neurotransmitters in vesicles. So these channels are for ions, and they activate a neuron by allowing depolarization. 107 says, after gene insertion leading to the introduction of a light sensitive channel protein into a mammal, which one of the following limits the cell types activated in response to light? Okay, so this is looking at what types of cells that these genes could be inserted into in the first place. And there's a sentence that describes that. It's the gene for such a light sensitive protein can be inserted into mammalian cells with the promoter specific for a particular cell type. And so the specific promoter limits the cell types activated in response to light. So think of it like this. There's a benign virus that has its genetic material that's been added to it, and it will go and be guided essentially by this promoter, and this promoter then makes sure that the virus can insert the DNA that you want, these genes that are useful here, into a particular type of cell. So then whenever you shine a light onto that cell, it will have that response. And any cells that aren't a target for this virus, and that will be defined of course by the promoter itself, they won't respond to the light because they haven't been given this gene, because the promoter didn't take the virus there. So this specific promoter will be what limits the cell types activated in response to light. So it'll be 107 B. And 108 then says, how does gene insertion lead to cell activity related to light? So let's go through the different answers here. A says cell metabolism is generally accelerated. So obviously this is going to be a set of channel proteins as I've drawn here, and it might have effect on the metabolism, but there's no reason to think from the information that we're given that it's going to just generally accelerate it. B says the vector distributes the channel proteins. So that's untrue, because the vector is what carries the DNA. So if you want to think of the virus, which I'll draw out here, so this is the virus and it'll have this stalk that will come down. If you hold the DNA in here, it'll effectively embed itself in the cell like this. Imagine this is going to be the membrane. So it'll sort of sit itself against this membrane here, and the DNA can pass through this stalk into the cell, and then it'll go into the nucleus and be expressed. So there's no channel protein actually being held within the vector itself, it's just the DNA. C says specific enzyme production by cells is increased. So these proteins are channel proteins, not necessarily enzymes. And then D says the cell produces and incorporates the channel proteins. So this DNA that's traveling to the nucleus is going to get expressed, and it's going to be turned into obviously this amino acid sequence, which will then fold up into these channels, and they will be incorporated back into the membrane here. So D is the right answer for 108.